So yeah, I was gonna focus on reproductive and immune systems today, and then maybe I don't think we're gonna get into much detail on these, so maybe we can do like a part 2 next week at some point before doing ecology, and then hopefully I'll talk to Tyson about a possible final review. Yeah. Okay, so just to start off with, I'm gonna give you a minute or so to look over these and try to match up on the left the type of cell to the description on the right. So let me know when you're comfortable with moving on.
Okay, I'm ready.
Okay, so yeah, let me know what you got. So, do you have access to draw and stuff? If you go to the top left.
Oh, like the pencil?
Yeah, the pencil. Do you want to try using the pencil and showing me what you were thinking?
Yeah, sure. I just had a quick question before. So do they produce the macrophages, right?
They produce antigens, and then what antigens do is they bind to the pathogens, and then that will kind of help the macrophages ingest the pathogens.
Okay.
So they help the macrophages do their job,
Okay.
but they don't produce them. You can go
I'm
ahead
actually
and.
not sure.
What did you say?
Oh, I said I'm not sure how to match them up.
Oh, that's okay.
Okay.
Is there anyone that you're definitely sure about? Or if you want a clue about one of them, maybe. I can give you a clue about the bottom two.
Okay.
So basophils, they're kind of they're pretty similar to mast cells. So if you remember what a mast cell does, then maybe that would help you. And neutrophils, they're pretty similar to macrophages. They're kind of like baby macrophages.
Okay.
Do you remember anything about? Yeah, perfect. Yeah.
What did you say about the basophils? Sorry.
They're similar to mast cells.
Okay.
Do you remember what a mast cell is, or?
I remember specifically what it does.
So here's another clue. Both mast cells and basophils, they have something to do with histamine.
Is that for inflammation? Kind of?
So have you heard of antihistamines?
Sorry?
Have you heard of antihistamines?
Yeah.
So when do people take antihistamines?
Is that the when someone gets like a transplant or something?
When someone gets a transplant? Yeah, that could be an example. That's
Okay.
perfectly. Another example could be when it's pollen season. So what do both of these situations have in common? What might happen? What kind of thing might go wrong during pollen season and during a transplant?
Okay, so the allergies, got it.
Yeah. So those kind of foreign substances in the bodies that cause like allergic reactions.
Okay.
Then to calm that down, you would take antihistamine. So you try to calm the effects of histamine, which triggers basophils and mast cells.
Okay.
So, some clues for the last couple. Humoral immunity relies on production of antigens, and cell-mediated immunity relies on production of cytokinins.
Is that like in the chart on one side and the other side, and then the one side focused on B cells and the other side focused on T cells?
Yeah, with the and then down the middle, it was the helper T cells, and then for the cell-mediated immunity, there were the cytokinin producing T cells, and then on the left for the humoral immunity, you had the B cells. Yeah, that's the one. Yeah, perfect.
Thank you.
Yeah, so you got them all right. Well done. And the starter might have given you a clue. We're starting with immune systems. So the first thing I wanted to ask was, why do you think B cell antigen receptors have two binding sites? So there are two places where they can attach to the pathogen. Like, why would that be? It's a tricky question, so don't worry if you don't get it. If you have the slides open, maybe take a look at, just as a clue, what happens when they start binding to what does it look like when they bind to these pathogens on both receptors?
They fit exactly.
They fit exactly. I have a picture here. Oh wait, it didn't switch slides. Okay, there. So do you see what's kind of, it's binding on, oh, you can't see where I'm pointer. Can you see now? Pointer?
Yeah.
Okay. So it's binding here, like if we consider this little Y shape over here, like one of the antigens, it's binding on this leg sort of thing, this binding site, to one pathogen, and then on the other binding site, it's binding to another pathogen.
Okay.
And then that pathogen is able to bind to pathogen, which is binding to another pathogen, and then so on. So if we like tried to imagine maybe that it just had one binding site. So let's take this antigen and say it just had one binding site. Then like this wouldn't be happening, right?
Yeah.
So we kind of have this little thing going on here. But because it has two binding sites, it can kind of like sorry, my screen's being weird. Can you still see my, nope, okay, wait. But because it has two binding sites, it's sort of making this like chain thing happening, right? What do you think the effect of that is if we like compare this to all of this, and it can be bigger, right? Like if you imagine like this was binding to something else and this were binding to something else, then it would be even bigger, right? So what's the difference between like what I drew there in blue and red?
Would it be killing more?
Yeah, perfect. Yeah. So like what's gonna come along and kill the pathogens that we were talking about earlier? Yeah, the macrophages are gonna come, and then how did they kill them? They
bind.
Yeah, they sort of like they swallow them, sort of like they ingest pathogens, right? And then they kill them. So macrophages are these large of Pac-Man cells that they come along and they eat the like pathogens. So if you have a lot of like if you were to just have, if we go back here in the blue, if you were to just have this little bit here and then another like little bit here and then a little bit here, like that would be so many pathogens spread out that the macrophage has to go and find all of them and eat them. But if you just had all of them kind of like grouped together in one spot, that would be very convenient to come and eat that one large group of pathogens at once, right? So it helps you get rid of the pathogens more quickly.
Okay.
Yeah. So do you remember these two terms, primary and secondary immune responses?
Yeah.
You can kind of guess, I guess, from the name, but yeah, what do you think?
I remember in the lecture he said something about primary is when like you're sick and then your body is getting used to having the pathogen in the body, so that like it lasts like 10 to 17 days, so like 2 weeks. And then secondary is once you have those memory cells in your
Yeah,
body, and the body's able to recognize them, and then it can kill the virus or bacteria quicker,
yeah,
or it
that's
won't
perfect.
occur. Yeah.
Yeah. So do you remember when memory cells get produced?
I think there's the memory B cells and memory T cells, really, or helper T cells, or something like that.
Do you already have the memory cells, like when your T cells and B cells are first produced, or what has to happen before you develop the memory cells?
I'm not sure. I don't remember actually.
So remember the slide we were talking about where there was like the cell-mediated response on the right and then the response on the left?
Yeah.
I believe the slide has kind of like arrows showing what triggers which process to occur.
Mhm.
So if you want, you can take a look at that and then see before what does it look like you have to have happen. So like I guess what my question is, do you have a memory cell before you encounter the pathogen?
No.
So then which response would it be possible to have both of these responses? Like, are they both options then if you don't have memory cells?
Sorry, could you ask the question again?
So if you don't have memory cells, what kind of response do you would can you still hear me? Sorry, okay. Um okay. Um it was so if you don't have memory cells, what kind of response do you have happening?
I think primary.
Yeah. Perfect, yeah. Um so why do you think so so when you have a primary immune response, that's the first time that you encounter the pathogen, right? So your cells have to kind of figure out what they're doing. And then what do you think the benefit of having memory cells is based on that?
I guess you get a quicker response and
Yeah,
stronger response.
yeah, so why would that be? Like, what's the difference between having a memory cell and not already having a memory cell? Like, what's the step that you're kind of skipping?
Your body will be able to recognize it from before.
Yeah, perfect. Yeah. So your body's already gone through this, it already knows what to do about it, sort of. So that's the benefit of developing these memory cells and being able to have a secondary immune response. That's what makes it quicker than a primary immune response. So how about then the first time you encounter a pathogen, but if you've already been vaccinated for that pathogen?
Mm. You encounter the pathogen then after you're vaccinated or before?
After you're vaccinated.
Oh, I think the vaccination has weak levels.
Sorry, you were cutting out. What did you?
A process of, well, I guess that normal process. I'm really not explaining it. Like, I think it's like prime, secondary.
Yeah, secondary. That's correct. So, how vaccines work, that the point of a vaccine is to expose you to the antigen. Sorry, so I've been saying antigen the whole time before when I meant antibody. Antibodies are what get produced by B cells, and antigens are the little parts of the pathogen. So when a macrophage engulfs a pathogen, the first like thing it does is it sort of cuts it up and it displays these antigens. So what the antigens are there like bits of the pathogen on the surface that your body can sort of use to detect the pathogen. Um, so the point of a vaccine is to be able to give you those antigens and give you a small immune response so that your body, without risking like really getting ill and actually contracting the disease, your body is able to develop these memory cells and make sure that when you encounter the pathogen, you don't have to go through the risk of a primary immune response because your body's trying to figure out what to do. So what if it doesn't figure it out? You want a secondary immune response if you can, like if you could response and already have the memory cells, because then your body already knows what to do. So it's more likely that you're going to be able to overcome that. Is that making sense?
Yeah, that makes sense.
Yeah. Okay. So for this next part, I thought we would try to test your memory of Bio 116. So last term, genetics component. So do you remember off the top the 4 different blood phenotypes? So the different blood groups?
There's type A, type B, type A, and type O.
Did I hear you?
Oh, I said type A, type B, type AB, and type O.
Yeah, perfect. So do you want to describe to me, or if you're not sure, that's fine, but are you familiar with the difference between the blood types?
Um,
And it's related to what I was saying about antigens.
Type A is blood with the antigen that's for A, and then I think it's the same for the rest of them. So type B can only accept antigen, and then type AB is the universal acceptor, so it can accept all 3. And then type O is so it can donate to the other 3, but it can't accept unless it's type O blood.
Yeah, so if I try to, 4 little blood cells. So you're right about, so they do have surface proteins that determine whether they're able to accept from different blood types or not, and give blood types or not, because you want to make sure that you don't have any surface proteins that your body doesn't recognize. So do you want to try to draw for me what you think they might look like? It doesn't have to be a perfect drawing, just to get an idea. You can make up any shape for the protein, like it doesn't really matter. But yeah. Just keep in mind which cells would have which type of proteins. So like if you have one shape for a particular protein and you think another cell has the same protein, be consistent with that. Or if you think it has a different protein, have a different shape. Or you can switch colors maybe if it's annoying trying to get the shape right.
Okay.
Do you see where you switch colors to the little circle?
Yeah. Yellow right
Oh, okay.
now. Thank
Oh,
you.
that's cute. Doing it in like 3D.
Oops. Is it like an eraser?
It's everything, but is it for the pink part? If you want,
Yeah,
we can
that.
pretend that that's a different type of cell if you want.
Oh, okay. Oh, well. That part too.
So you can draw arrows to which type of cell it would be if you want.
Okay.
Maybe draw them in like, is there a green? Yeah. Yeah, so this first one, on the very left, would that be, so like, wait, let me use my, this, what type of blood group would this be, do you think?
I meant for it to be type A, but like I drew a purple part, but that would be like A blood group. And
So you're gonna ignore the purple and then it's A. Oh, that's what you meant. Oh, sorry. Oh, fuck. Oh, sorry. Sorry. Okay. Sorry, I didn't see that you'd drawn the purple on the third one, so I thought that we were gonna like make them different cells, and then I accidentally erased everything because I was trying to undo my pointer.
that's
Sorry,
okay.
I can do that, or you can redo that. It's up to you. I just feel guilty now if I've undone your hard work.
That's alright. I can still draw it.
Thank you. Sorry.
I have a question. So for the exam, would you like ask us to draw these or no?
He wouldn't ask you to draw these, but I think it's helpful to understand conceptually why this is happening, which is, yeah, I guess I kind of like the way I've been designing the sessions is like more for conceptual understandings than
Uh-huh.
that means is like sometimes we don't go into that much, so we haven't gone into detail about everything he's talked about, um because I think that you guys are kind of like well practiced in learning how to memorize things sort of, but
Uh-huh.
have a conceptual understanding that makes it a lot easier to answer. In particular, Dr. Nelson, I think, likes to write a few questions per exam that really make sure that you conceptually understand the material. So he might give you
Uh-huh.
like a hypothetical scenario, and then you'd have to kind of use your reasoning and knowledge of the course to work out what would happen. I'm not being very clear there.
Oh, no, I
Um,
know what you mean.
I can't remember exactly what the question was, but I was in his cell bio class last term, and there
Uh-huh.
was a question on the final about what how you would design a hypothetical treatment for a particular condition, and we had to use our, but you couldn't really, there's nothing you can memorize there because it's sort of like a hypothetical question, but you had to kind of be aware of which processes would matter in that situation, which wouldn't, based on sort of a more conceptual understanding of the course material. I'm
Okay.
sorry, it would be more helpful if I didn't, but
Well, that's alright. Thank you. So, like
okay.
for our final, is there going to be a written response or is it still just all multiple choice?
I think what he's gonna do is he's gonna write multiple choice questions until he feels like there's enough of those, and then if he feels like um it would be appropriate to test with a written response question, he might have that. But I think especially because it's online, that's gonna be a little bit, he might not, that would be like very difficult to grade for like such a large class if
Uh-huh.
like these short essay questions. Like it might be, I'm not really sure how the automatic part of the Canvas quizzes work. Like if you can have like maybe a labeling question, that might be like for the male and female reproductive organs, you might have a labeling question. Um, and that wouldn't be like crazy to have in a large class. He might go for short essay questions and have and grade them all, but I think so my final exam last term was all multiple choice. We did have short answer questions on the midterms, but he just happened to not write them for the final. It really yeah, I wouldn't really study based. I guess you should study to an extent based on the type of the question, but if you were to do that, expect multiple choice, I would say.
Okay.
But for studying for the final, I would really recommend just making sure that you go through the final exam review sheet and you really answer all of those questions in as much detail as he's provided in class. Like if he's mentioned something interesting about it in class, you want to remember like the way he's talked about it, like the connections he's made using that. Because that would come up on the test because it really makes sure that you were paying attention, right? Instead of so why he likes to test on that.
Okay. All right. Thank you.
Yeah. So yeah, walk me through. So what we want to do is we want to think. Then so let's look at AB to start off with. We want to say like. So you said this was the universal acceptor, right? So it can take from any. Yeah. So why would it be able to take blood from B?
Because they antigen receptors, I think it's
Yeah,
called.
they proteins on their surface. So they both have these pink proteins on their surface. So if it were to take blood from B and it was inspecting this blood sort of, it wouldn't be seeing anything foreign. It would see these pink proteins, but that's normal because the body already has pink proteins on the blood. So that's it's not worried about that. So walk me through why it would be able to accept from A then. You can use a pointer if you
Oh, okay, I see that. Because the same with A, it has the same proteins as AB.
Yeah. So it has these yellow protein. Yeah. Oh,
Oh, there
sorry.
it is. Okay.
Yeah.
One there and there.
Yeah. So then the body wouldn't think that's foreign. Is it able to accept from O?
AB?
Yeah.
Yeah, it is. Oh, oops, I forgot.
You can maybe cross out the orange protein if you want. Do you want to walk me through why you're crossing out the orange protein?
Because type O blood doesn't have the same proteins as A, B, and AB.
Yeah.
Always.
Adding it. So you're adding an orange here. Okay.
Wait, would it make sense to add orange onto A, B, and O?
So let's have. Ah. So this is tricky because it almost would, but okay. So talk to me again about which blood types can A and B accept blood from?
AB. I mean,
Yeah, themselves. Is that it?
and O.
Yeah. So you said the O was the universal donor, right?
Uh-huh.
To A, B, but it can also donate to B and to A. Then what does that mean? So right now is B able to accept blood from O?
No.
So how can we fix that? Maybe on your drawing. It would. And what about A? Yeah. Okay. So now, yeah, that's perfect. So now we've um now everyone that's able to accept from someone has the same proteins, and everyone that's not able to accept from someone doesn't have all of those proteins.
Uh-huh.
Yeah. Okay. Um. So that's perfect. Yeah. So the way that I like to think of it, Are you still here?
Oh yeah, yeah. That was
Sorry, a
just
weird sound happens like every time I switch slides, and I got scared that you're like, so the way that I like to picture it is I like to picture these tiny little A's and tiny little B's on the cell. So if the name has an A in it, I'm gonna give it an A. Oh, let me get my pointer. If the name has an A in it, I'm gonna give it a little A protein. If it has a B in it, I'm gonna give it a little B protein. So this has both, so I'm gonna give it both proteins. This doesn't have an A or a B, so I'm just not gonna put any proteins on. It's the same
okay.
thing as adding an extra O protein to all of them.
Mhm.
Conceptually, it's the same thing. But I don't know. This is the way that I like to remember it, but I like your way too. And I like that you were able to figure that out because that was tricky. So well done. Um,
Thank you.
so we kind of already went over this. Yeah, I'm proud of that. No, I'm just kidding. But yeah, so do you want me to, or how about you walk me through using this drawing? Who can accept blood from whom? So you kind of already did it, but um yeah, if we draw a table out. So I'm gonna draw a table whilst you start.
Okay. So blood type A can accept from blood type A and blood type
And um explain why as well.
because blood type the pro has proteins that are specific to that group, I guess. So then blood type O also has no, it doesn't, sorry. Blood type A also has the proteins for blood type O.
Yeah. So
And
yeah, try to reference the diagram and explain, is it seeing anything for like walk me through. So it can accept from A because A has all the same proteins, right?
yeah,
Walk me through the next one, B. What would be the problem? Like, what is it seeing on B that it's getting upset about?
for blood type A.
Yeah.
Um, if blood type B was to accept blood from blood type A, it wouldn't work because the receptors on B work with the ones on A, I guess. So then the antigens might be able to bind to the receptor, I think, something like that.
So
They don't have to
what would be happening is kind of the same thing as what happens when you have like a pathogen in your body. Like the body would, so let's pretend that you're saying if B accepted from A or
say.
tried. So
Yeah.
then this person who's only seen, their body has only seen these B proteins, all of a sudden there are these weird looking A proteins. So like what happens when, like how does the body know self from not self? It detects on the surface that, I've never seen these little proteins before. So it kind of thinks that this is a pathogen, and these become the antigens, and so the body starts attacking. It treats these cells just like pathogens, and it starts attacking and killing these cells. Um, because it's never seen those proteins before, so it looks like. So remember, Dr. Nelson was talking about the most basic part of an immune system is recognizing self from not self, because the point of the immune system is to attack not self.
Yeah.
So, and to not attack yourself, that would be bad. Um, so this person who's only ever seen these B proteins sees these foreign not self A proteins and then starts attacking them. So yeah, so you're right, that would not work out. Yeah.
Okay.
Um yes. so just pick another combination, I guess. We'll just fill in this table.
Blood type AB would be able to recognize blood types A and blood type B.
So why would that be?
Because I'm not sure the terminology to use. Like I always get the like with the antigens and
you can just call them A proteins and B proteins and self and not self.
okay. Because blood type AB has A proteins and B proteins, so it recognizes it as itself, so it's able to accept blood from both of those.
Yeah, perfect. Yeah. So
and
we
then,
have.
Oh, you go.
No, it's fine. I was just like that we have. I was just saying what we had left to talk about, but that's you can see anyway. Yeah.
Okay. So then blood type B also the A protein, so it's able to accept. And can it accept from AB or no? Because it has.
So is it recognizing every single protein on AB, or are there some foreign proteins?
Oh yeah, there's some foreign proteins.
Which ones would those be?
The A.
So then would B be able to accept from AB?
No.
Oh, yeah.
Okay.
Would think it's not self if it sees the A.
Okay. And then type O doesn't have the A proteins or the B proteins, so it's not able to accept from either of those.
Yeah.
And then, yeah, I think that's it.
Yeah. Maybe you can just tell me the answers to these.
Oh, to the.
If you want.
Oh, which part, sir?
So the table over here.
Like X's and stuff.
Yeah, with X's and ticks maybe.
Oh, okay. There would be an X between right here, and then there would
What should I put here?
a check mark.
Okay. Yeah.
And then there would be a check mark here as well. And then a check mark. Well, just between the A and AB part, there would be
Sorry, what would there
and
be?
then a check mark between
So is A able to
AB.
accept from?
Oh, I didn't see that. Oh, I messed it up. I read it wrong. I think there would be an X there.
Yeah. Why would there be an X there?
Because the blood type A is not able to recognize the B protein, so it's used as non-self.
Yeah, perfect.
And then for O and AB, there would be an X.
For O and A, so here?
Yeah, on the bottom, yeah. And beside that, there would be a check mark for O and O. And then for all of those above, I think they'd be too.
Could you explain why?
Because blood type A, B, and AB have proteins for blood type, but then blood type O doesn't have those recognizable proteins on itself, so it's not able to accept from blood types A and B.
Yeah, perfect. So there's nothing foreign on O to those blood types. Yeah. Yeah, you got them all right. Do you remember what the difference was between a genotype and a phenotype?
The genotype is the gene on it, yes, and
Yeah.
then the phenotype is the physical characteristics.
Yeah, so the genotype would be like writing out which alleles you have, and then the phenotype is like, okay, but then how does that information translate into the actual protein? Like, what does the protein do? What does it look like? So do you want to start out with identifying the alleles? So for you know how we usually use one letter for an allele, and then we have a capital for the dominant and a lowercase for the recessive type. So
Mhm.
for blood type, you use I, but then we go back here. There are four different blood types, right? So we're kind of, when you're thinking of alleles, think of like they need to make certain proteins. So let's think of these proteins. How many different proteins do we need to make?
Uh, Wait, sorry, could you re-explain that?
So when we're deciding which alleles we have, we want to say which proteins do we need to make. So which proteins are we going to be making? Like on what does it look like we're making on the surface? How many different types of proteins?
Would it be one for each or two?
So two total, and those would be
I, A, and IB.
yeah, perfect. Um, and then what would the recessive case be?
The little I for blood type O.
Yeah. So the way that I like to remember it is so what this is called when you have like two forms of the capital letter and then a superscript is
Mhm.
codominance. So what that means is that if you have both of these, it's not like one is more dominant than the other. You kind of do both things. So this is always recessive. So if you have like a dominant allele and a recessive allele, the dominant one always wins out in the phenotype. But when you put together two co-dominant alleles, they kind of like they create like a completely new phenotype together. So that's what makes them co-dominant. Like one isn't dominant over the other one. So I like to
Okay.
think in terms of this. So the IA is like adding the little a proteins, and the IB means adding the little b proteins, and then the recessive lowercase I just means like your kind of default base case. So you're not adding A or B protein, so you just have this little circle. You just have this O blood type. There's nothing special being added there. Nothing extra being added there.
Okay. I have
So
a question.
yep.
Could there be big IA twice, or is it always IA little I?
So let's think about that's a good question. So if we had IA twice, what
Mhm.
do you think is our genotype? What do you think the phenotype would be? So what would each of these alleles be doing? And then let's compare that to IA and then lowercase I. So
Um,
what kind of proteins are you making in this case?
just the A proteins.
Yeah, we're just getting um we're just getting these little A's. So what blood type is that?
Blood type A.
Yeah, so this is type A. Oh, I can't type A. There's a text option, but I'm just too lazy to like switch in between the two. What about here? So I'm gonna draw a little blood.
Would that
So
still be blood type A?
so walk me through your thinking. So what happens when you have a dominant allele and a recessive allele?
The dominant shows, but the recessive is still there. It's just not visible. Yes.
Yeah, and I feel like
Recognizable.
blood type example makes it very clear because this is how I like to think about it. So I like to think about this allele. It says make A's, so that's what it says, and then this one just doesn't say anything. So then all is that you've made these little A proteins because the other allele told you to do it. And you can't really tell that the recessive allele is there because it didn't tell you to do anything. So this would be blood type A, right? Put together two of these recessive alleles.
It would show the blood type O, so just a circle.
Yeah. So did we add anything to our blood little blood cell?
No.
No. So I would say that that's why I feel like this example makes it very clear like what it helps you understand like the difference between recessive and dominant because this one's not really it's this isn't a perfect it's not like a perfect way to think of dominant and recessive alleles, but I just find it helpful. Like it hasn't done anything, so that's why you didn't see it.
Okay.
So I don't know. That's just the way I like to think about it.
Yeah, that was...
So how about you try using that to write out, maybe you can use the text option, and then I don't really mind if the formatting is weird. Like if you write it like this, like I still understand what you meant, like that this is capital, like opposed to this. That's fine. Do you want to write under each of them, like, or not under each of them? Because let's write up somewhere the different blood types. So I write it here, maybe. So A, B, A, B, and we put O here, and I'm running out of space. Yeah, and do you want to maybe just write out somewhere here what you think the genotypes would be? So the IA allele makes you produce A proteins, and the IB allele makes you produce B proteins. What do you think the AB blood type corresponds to if it's producing both?
Oh, what do you mean by what does it correspond to?
So you see how we wrote out genotypes? Yeah.
Since there's the big I for both of them, both alleles which should be present and show.
Yeah, so do you want to write that out? Yeah. Thank you for adding the explanation. That's okay with just the genotype is fine. So
Oh,
do you
I
want
thought you read the explanation. Sorry.
Don't worry about it. That's fine. That's perfect. Thank you. So let's try doing that for the other blood types. Yeah, great. Perfect. What about this? This is a possible blood type. So where did we see say that that would
Oh
go?
yeah. Is it possible to? Oops, I made a mistake with that one.
That's okay. You can draw arrows to where you'd like them to be, maybe.
Oh, I don't know. For the AB, is it possible for it to be IA little I and IB little I? Or
So how many alleles do you have of a gene? You get like a set of alleles from your mom and then a set of alleles from your dad, right? Or from your father and mother. so so then how many alleles are you gonna have of the same gene?
would it be two?
Yeah. So the reason why that wouldn't happen, like if it were to happen, like if you added all of these alleles up, then that you would still get an AB blood type, but it's just that you can't have four alleles, or in humans you can't have four alleles of the same gene.
Okay. So would it just stay the same?
What do you mean?
Like, um, like how would I add the little I to it? Because if AB
Yeah,
blood is equal
kind of
to,
big, right? I'm
oh
just
sorry, I
drawing.
didn't hear that. Like,
Um, so
oh,
you only have two copies of the gene.
mhm,
So you can only have two possible, or you can always only have two alleles. So if we want, we already need to have both of these alleles to get this blood type.
mhm,
Wait, let me use my pointer. We already need to have an A and a B allele in order to produce the AB blood type. So
mhm.
there's no ways to add any recessive alleles because we're already using the both of the codominant alleles. Does that make sense?
Kind of. So like, do you mean like, so the co-dominant alleles would have the little I on it, like at that part already on it? Is that what you mean?
Yeah, so in the case of blood type, the AB, like the little I for blood type, so the blood type, they're not adding any extra proteins on.
Mhm.
So all of the other blood types, they already have those proteins. Um, they're just adding some extra ones.
Okay. Yeah, that makes sense. Thank you.
Okay. So yeah, you got them all right. So I was going to move on to reproductive systems. Um, I know we don't have a lot of time left, but do you mind if we go over time?
Yeah, sure.
Okay, great. Okay, so we're gonna start off with a quick true or false.
True. I don't know.
So so we do have in most cases genetic I can't see what I'm typing because it's in white. Hold on. I'm just gonna start typing from the beginning in black so that I can see. We do have in most cases genetics. So that was the abbreviation for that was GST. Do you remember if there was a different abbreviation for a different type of sex determination?
Oh no, I don't remember it.
So I'm gonna tell you the abbreviation, and we'll see if it jogs your memory. So it was TSD.
Trait? I don't know. I guess trait.
What did you say?
I said traits.
I'll describe how it works, maybe. So the way it works is the temperature at which so organisms that have this type of sex determination, the way that it works is the temperature that the eggs are grown in, so they're incubated in, will determine the sex of the hatchling. So a benefit of that Dr. Nelson was mentioning in the lecture is that the parent can kind of control the sex ratios of the offspring by controlling or by selecting environments of different temperatures for their sex. So is this making sense?
Yeah, I remember this.
Do you want to guess what the name was?
Is it the temperature sex determination?
Yeah, pretty much. So yeah, so always determined by genes because sometimes it's determined by temperature, and that's called temperature dependent sex determination. Might you remember an example of an organism that that does this?
Was it ice?
Not quite. It's not flies. I don't know why I said not quite. I was just trying. This is just sort of sorry. This is just sort of something you have to memorize. Yeah, it's just So he was saying in the lecture, turtles and alligators do this.
Okay.
Maybe there's something else I don't really. I just remember that turtles and alligators do it.
Okay.
I don't know if that's quite something that he would test on, is like specifically what organisms do this, but it's just I don't know. Turtles and alligators, they're kind of easy names to remember compared to some of the other, like you have to remember that nematodes do this, or that,
Yeah.
I don't know. That's just something that I remembered. Okay, next question.
Is it egg cells?
Yeah. Can you tell me why?
I think he said something about how sperm is cheap and it's easier to produce, but then egg cells, on the other hand, takes a lot more investment because of the organs that come in with them, and like just something like that. I'm not sure. How eggs are more expensive and stuff.
Yeah. So yeah, I like that phrase of more expensive and cheaper, because that's kind of what we're talking about. So if you want, you can use the human kind of reproductive system to explain why more investment goes in producing egg cells than sperm cells.
I guess egg cells they're produced once a month, or like
Yeah,
every,
perfect. Yeah.
and then sperm cells.
Yeah, pretty much. Yeah, they're being pretty much constantly produced. The other interesting thing, so how many egg cells are produced once a month?
Just
in
one.
humans. Yeah, just one. But remember, so if we do meiosis, so let's say we did meiosis 1, so that the first half of meiosis 1, I can't type like, forget, just take my word for it. So how many cells do we have after meiosis 1?
2.
Yeah. And then we do meiosis 2, and then how many cells do we get?
4.
So out of these 4 cells, how come we only got 1 egg?
I don't remember exactly, but I remember there's like a word for it. It's like oocytes or like oocytes or something like that. I'm not sure
Yeah, so
exactly.
this is the process of producing egg cells. And the oocyte is the egg cell, so the actual egg cell. And what happens is actually, because there's so much reproductive investment into that one cell, one of these cells is, we're gonna have these little wimpy other cells, and then one giant egg cell that took all of the cytoplasm, and all of the investment went into making that cell. Like, we don't really care about the other ones. The other ones, they actually, so they're called polar bodies, and they just like, we just get rid of them. We don't really care, because reproductive 4 cells of reproductive investment goes into that one egg cell for the entire month. So yeah, that's another. I feel like that's the kind of, it's a pretty funny example, because it sounds like such a waste. It's like you could have had 4, and you ended up with 1, but it's because you want to use up all of that cytoplasm and like really pack that one egg cell, and just put all of your effort into that one egg cell. So much effort that it could be 4 different cells. I just think that's pretty funny. I don't know, to me.
Easy to remember.
Yeah, easy to remember as well. Yeah, the other thing is, so once the cell is fertilized, what's gonna happen? Yeah, like, which one kind of lasts? Like, does the sperm cell? I'm trying to draw a sperm cell and an egg cell. So once like fertilization happens, so we have the genetic material being passed on, and we actually also have the mitochondria from the sperm cell passed on. But then what do we do with the rest of the sperm cell? I don't think it's required for this course. If you don't know, that's fine. But like, the rest of the sperm cell is just sort of discarded, and then it's like everything else you get from the egg cell.
Okay.
And so is it the parent that housed the egg cell, or the parent that housed the sperm cell that the that will where it will house the zygote?
Depends on the organism, because I remember there's this one organism that like the mother produces the egg, but it's kept in the male. But then I think it's like the 3 different mechanisms for birth. It's like
Kind of,
ovoviviparous or something.
yeah, so there are mechanisms where the zygote will become nutritionally independent and even leave the parent. But when, so after, immediately after fertilization occurs, and you like first have the zygote, which parent is it gonna be in?
Oh, the mother.
The one with the egg cell. So I guess like that's another way to think of, all of the reproductive investment is going into the egg cell, because eventually like that's where you want the zygote to develop as well. I guess. I don't know, the other example was clear with the 4 cells, but yeah. Yeah. Perfect. So this is gonna be tricky to remember, but let's see if you
forgot the words that were used for it, but like there was this chart, and then they're all female because they produce eggs as much as they can until they run out, and then when they it produces a haploid, I believe.
Oh, so
And
that was for rotifers. So what you're thinking of is mixus. Um, it's not quite the same thing, but um, I think he talked about it right before he talked about mixus. If that helps you remember.
oh no, I don't remember. Sorry.
So that's okay. Don't worry about it. That's fine. I said it was a difficult thing to remember. So do you think these lizards, if they're all female, do you think that they do sexual reproduction or asexual reproduction?
Asexual?
Asexual. Do you think that? Or I guess it's kind of obvious, but
Mm, I'm not sure, actually.
that's fine. So sexual reproduction, kind of relies on having like producing male and female gametes, and then producing offspring like through the fusion of the male and female gamete. Um, but if you don't have male lizards, you're not gonna make male gametes, right? But are they gonna make female gametes?
Yeah.
Yeah, so they're gonna. So this is what we know so far. We know it's asexual reproduction. Wait, I'm just gonna type it. Sorry. Asexual reproduction, and we know that they produce egg cells. So egg cells are usually produced for sexual reproduction, right? So there is a word meaning producing egg cells and mimicking sexual reproduction, but in order to do asexual reproduction. It's a pretty like long word. So if you don't remember it, that's fine.
Okay.
It starts with a P.
Parthenon.
Yeah, perfect. Yeah, I got it. Parthenogenesis. Yeah. So in summary, what was that? Try to summarize what we talked about. So to define parthenogenesis.
Reproducing without a male, I guess.
Reproducing
Asexually
asexually without
a male.
yeah, asexually without a male, specifically by using by producing what type of cells?
Egg cells.
Yeah.
Okay.
Yeah. And I think Dr. Nelson went through a couple examples of how this could happen. Like you could have let me try to get the right color. You could have a diploid organism, so a 2N. So difficult to write with this. Making little 2N eggs that can just develop by themselves into, like, they're basically already the zygote. Or you could have 2N organisms producing 1N eggs that fuse. So you have like the eggs fuse together to make the 2N zygote. Or you could have this was an interesting case where sometimes you have a tetraploid organism, so the organism itself is 4N. So then when it does meiosis, then the number of chromosomes divided by 2, it produces a 2N egg, and then that egg is like a different kind of, it becomes a different kind of organism that's 2N. So yeah, there's different ways that this can happen, but what they all have in common is that they're asexual and they involve production of eggs.
Okay.
So why are these? So we talked about when asexual reproduction and sexual reproduction are used, and it seemed like generally asexual reproduction is for sessile organisms. So if you stay in one place, because if you're motile, then you're able to like find a mate. If you stay in one place, do you remember his example about finding a date in high school or something?
Yeah, I remember that.
Yeah. So something about a car and moving around. If you move around, you can find a date. But if you're stuck in the same place, then you can't find a date. So if they can move around, then why don't they just find someone to mate with? Why do they do asexual reproduction?
Um,
It is tricky, so if you don't remember it, don't worry.
uh, something about having the same species reproduce over and over again. I forgot what he said exactly, but it was like,
Yeah, that's a good point. So that was about so if you were able to survive and nothing's changing in your environment, what's different about doing sexual versus asexual reproduction? Like what would happen
um,
to the next generation if you did asexual reproduction versus sexual reproduction?
they would stay homozygous, heterozygous.
Yeah, that you're kind of approaching this homozygosity because you're just giving them the same alleles over and over. Um, and so like if there was nothing wrong with your alleles, like if they worked in that environment and there's nothing changing about the environment, then there's no you don't need to fix it if it isn't broken, sort of thing. Um, so then that's a good point. There was something also specific about these lizards and something else like they can move around, but it's still hard for them to find a mate for a different reason.
I'm not sure.
Yeah, it's pretty tricky. I'll give you. I'm gonna give you the answer, and then I want you to explain the answer. So there's not a lot of lizards around, so there's a low population density. Why would that make it difficult to do sexual reproduction?
There's not very many males since they're so used to reproducing sexually.
Yeah, so there aren't that many other lizards for them to mate with. They can't find another one that easily. So they can't rely on sexual reproduction because if they don't find a mate, then they just die out, and it's pretty tricky to find a mate because there's so few of them. So it's sort of like there's not enough people to mate with for their chances to be high enough. So it's just easier to do asexual reproduction. Yeah, perfect. Um, so I might come back to the next slide at the end. Let me decide. I'm gonna come back to the next slide later. I'm gonna skip this one and just go to this one.
Okay.
Do you
I remember them, but I don't remember what they mean.
So I'll give you a clue. They're related to polychaetes. And do you remember what polychaetes are? Or an example of a polychaete?
No.
So polychaetes are these um they're these segmented animals, like for example, a tapeworm is a polychaete, I believe. I'm trying to draw a segmented. And so what do you remember is weird about polychaete gonads? So they're sex organs. Can I like copy and paste No, no, I'll have to draw it again. I'll draw it again and then give you a clue. Um, sorry, it turned out different looking. It's fine. See, I'm too lazy to draw the segmentation, so just pretend that that's.
Mm.
So if I were to tell you that this one is the atoke and this one's the epitoke.
Oh, is it? You said gonads, right?
Yeah.
Is it atope without a gonad and then epitope with a gonad to
Yeah,
like
perfect.
reproduce when they want to?
Yeah, so they start off without even having the organs they need for reproduction. And then so that's the atoke form, and then the epitoke form is when they develop the organ that they need. So when they become reproductively mature. Um yeah. So they don't even have gonads to start off with. They start off as this, and then they become this when they're ready to reproduce. So that's the interesting thing about polychaetes. Yeah. So we're nearing the end. Do you remember what a pedipalp was?
Yeah, I think it was the I don't know if it's like a structure or something, a spider. It's in the middle, I believe, and they use it to hold sperm
Yeah. So
until they're
what did you say was the last thing?
until they're ready to reproduce.
Um. So that you're right about the male spiders using it to hold sperm. Female
Mm.
spiders also have this, but they don't use it to hold sperm. They don't produce sperm. That's what the male spider does. Um, they don't. So do they hold sperm in there for longer term or for shorter term?
Uh, Shorter term
Yeah. Where is the sperm produced?
and
Try to draw a spider.
testes.
Um, think in terms of the like segments of the spider. This is so ugly. Oop.
Um, the name of the structure.
Um, so think stomach in humans maybe would help you.
Mm.
The word for stomach, muscles on the stomach. Think, and I'm gonna draw a pedipalp. I'm gonna make it kind of bulbous at the end to hold the sperm. Make it look funny. Um, do you wanna maybe indicate with your pointer on the drawing?
Where it is?
Yeah.
I think it's like there.
So a little bit lower. It's like in the abdomen is like where it would be produced. And so the spider, it's funny, the spider releases silk and then releases, I'm gonna pick out another color first. I'm just picking yellow randomly. Releases the sperm on top of the silk. And then it uses, let me pick blue. And then it uses these thingies, so the pedipalps, it uses them to like suck up the sperm that it just released. Um, air, and then that's what it's gonna use to fertilize the female spider. So I'm gonna draw another spider. Um,
I remember in the online lecture he posted, he said that females have their own pedipalps and that like
yeah,
the male one is more rounded. What does the female one look like?
It's kind of just like it looks like the spider leg, but like smaller, and then the male one will look like weird. I don't know. It's like I feel like it's a subtle difference, but I don't know.
Okay.
Maybe I get distracted by spider legs and I don't really notice anything else. Um, so you have like the opening here, and then you also have this like structure on top. And do you remember what that structure was called?
Mm-mm.
I'm gonna do a kind of like it was called. Wait, let me count the letters. 1, 2, 3, 4, 7, 8. Do you wanna guess? I don't know.
Mm. I don't know. Epigynal?
Yeah, perfect. Yeah, epigynum. So P and then I. Epigynum. Um, and then so what the male spider does is that. So what do you think it's gonna do? Like now it has the sperm and the pedipalps.
They discharge it into the epigynal.
Yeah. So yeah, that's the first way it helps. Um, and so do you remember that Dr. Nelson was saying that male spiders are vulnerable to like sexual cannibalism. So they're vulnerable to being eaten by the female partner if she's feeling hungry.
Oh yeah, because they go closer to, I don't know what part of it, but they go close to one part and then it's easier for her to eat them.
Yeah, the video he was showing, he was saying that this spider is vulnerable because the other spider has her, like, she's right on top of the abdomen. So it's like a good angle for like eating him and you can't really do anything about it. Um, and he was saying that like, it was funny, he was kind of saying like, spiders will the male spiders will try to do will have different adaptations to help them like not get eaten or not really help them not get eaten. It's okay if they just delay not getting eaten until their sperm is fertilized the egg. So like until they've gotten like once they're done with the job, they don't really mind getting eaten. Like that's just something they have to deal with. And then it was, oh, but like it's not just about getting the so what does the epigynum do? So
um,
once the sperm is in there, does it immediately like has fertilization already occurred or
I think the female can hold
yeah, perfect,
she
yeah.
wants to meet with someone else or if she decides to.
Yeah,
Um,
so
yeah.
the female spider can hold it for up to a year and then you said if she decides to be with someone else. So yeah, if she finds another male spider, um, and just decides to get rid of the first one's sperm, she can just ditch that sperm, and then the other guy got eaten and didn't even get to pass on his genes. So they have to figure out a way to make sure that once the sperm's in there, it's not gonna get ditched for someone else's sperm. So how do you think the pedipalps might do that?
I think he said something about them breaking it
Yeah,
off.
yeah. So this is like I don't know. It sounds really funny. So they like break off this body part and insert it into the epigynum to block the epigynum off.
Then they have no choice.
Yeah,
It's so savage.
yeah, Yeah, I thought that was interesting. So before the last question, I just wanted to try to go over all of these terms, because they've been coming up a lot, and I feel like they're good to like really, really understand. Um, and yeah, I don't really have. We can just like this is a little less structured, so we can just like kind of talk about what your understanding of them is and how to remember all of that.
Um,
So, yeah, you can start wherever you want.
I don't know I don't remember gonochristic or Viburnus and Protandrous, but I
That's okay.
remember the first three from like the first half of the semester when he taught it to us.
Yeah.
But I was kind of confused because like there's like different meanings
Yeah.
towards them.
Animals and plants. So let's focus on animals. They're pretty similar, but let's focus on animals. So monoecious. Do you want to define that for me?
I know it means like one house, but I'm just gonna guess it's houses only one type of reproductive structure, so like male or female.
Yeah, that's exactly it. Yeah. One type of um like 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 all of the organisms have the same type of like reproductive organs, you said, yeah. So you don't really have like a male and a female sex. They're just all, or like you don't have multiple sexes at all. You just all have the same thing going on.
So is it like in one population or like group?
It's like all organisms of that species
Okay.
have to have the same reproductive organisms.
Okay.
What about species?
They can either be male or female
Yeah,
in that
so
order.
dioecious means you have two sexes. So you have um organisms can have one of two different types of reproductive organs. Um, and so there are organisms that, or there are species that don't fit into either of these two categories, that have, they don't all have the same thing, and they don't all have one of two, like they have multiple sexes. Um, you don't need to know a word for that. You just need to know these two words. So mono one, dio or di two. So either mono is like not really, don't think of it in terms of one, maybe think of it in terms of like the same,
Like all
like
kind of.
monochromatic means the same color or something. So yeah, mono in terms of like they all have the same. Dio, di in terms of like there are two possibilities.
Okay.
Um, and then hermaphroditic. What did that mean? If an if an organism were hermaphroditic,
Um, is it both reproductive structures?
yeah, so they have both the male and female reproductive structures or reproductive organs.
So is that also in one, like in all organisms in that group?
Yeah, and then here's where it gets complicated. You can be hermaphroditic and still be um dioecious by being gonochoristic. So let's think back to the rotifers, right?
Mm-hmm.
Remember you were saying that they usually like are female, and then sometimes they can um turn to the haploid generation and become male. So then the same organism can be hermaphroditic. Like it it can be both. Or
So like
actually, now that I think about
back and forth,
it,
kind of.
sorry. Actually, now that I think about it, they're not hermaphroditic. Um, but pretend that they were able to be. So what would that require? Like to be able to. The reason they're not hermaphroditic is it's not the same organism becoming male. It's like making. It's undergoing meiosis and then producing a haploid generation that will produce. Yeah, it's not the same. Okay, don't think of that. Never mind. But just think of what would be. So if you're hermaphroditic, so the like every organism can be both male and female, but there are still two sexes. Then what has to happen? Like, can you? Is this making sense? Not really, right?
Kind of, yeah, just a little. Mm,
So I'll give you a clue. It at some point you have to be able to have both male, like you have to have a male reproductive organ at some point and a female reproductive organ at some point on the same organism. At some point is the key word, the key phrase.
mhm.
So then what do you think gonochoristic means?
Um, at one time they can only be that certain
Yeah,
sex.
perfect. Yeah. One sex at a time. So then you can be male and then become female, female and then become male, and then you would still be dioecious, but you would also be hermaphroditic. So what that's called that so what that's called is um being a sequential hermaphrodite. I'm sorry, sequential hermaphrodite. So sequential hermaphrodite means that there's like a sequence of sexes. Like you either start as female and then become male, or you start as male and you become female. Um, and do you remember what the other type is called?
Mm-mm.
Um, so it has to do with synchronization. That's a clue.
I can't remember.
Um,
Am I allowed to look at my notes?
yeah, okay, yeah, we're almost done.
Okay. Mm.
So it has to do with synchronization. It sounds like the word.
Oh, synchronous?
Yeah, so it's both at the same time, so it's like happening in sync, sort of, I don't know. Synchronous hermaphrodite. So that's when you have both of them at the same time. Sequential is when you have one and then the other. Um, so are you familiar with like the prefixes like andro and gyno? So what do you think gyno has to do with? What does gyno kind of sound like?
Gynecologist?
Yeah, gynecologist. Who do they work with?
Females?
Yeah. So people with like female reproductive organs. So like protogynous, does it sound like sorry, I'm gonna plug my Mac because it's 5%. Protogynous, does it sound like you start off as like which sex does it sound like you start off as?
Okay.
Yeah. And then andro um, proto-androus. What does that sound like?
Um, the opposite.
Yeah, so you male. So proto-genesis start female, become male, and then proto-androus start male and become female. And he was giving us an example. I'll give you a clue. I'll try to draw it of organisms that are proto-androus, and it had to do with a Pixar animation. The example he was giving
Oh,
us.
it was Nemo.
Yeah, Nemo. What was he saying? Do you remember?
I think it was like if it's Proto Andrus, it was like whichever male is biggest at the end has
Yeah,
to turn into a female.
yeah. So then you have like a group of males being born, and then the most dominant, I guess, like the largest male becomes the female. This is interesting. Yeah. Yeah, perfect. Are all of these terms making sense?
Yeah, they are. I didn't really understand it when he like taught, like when I was listening to the lecture. So that
Um,
really
so what if I were to ask you, sequential hermaphrodite, is it dioecious or monoecious?
um,
So this one's the sequential one that so it starts as female and then it becomes male.
oh,
So
that is just
yeah, why do you say that?
because it's switching between.
Yeah, exactly. So it has one sex at a time, but it has two possible sexes. And then a synchronous hermaphrodite is when they all have both reproductive organisms at the same time.
Okay.
So then what are they?
Hermaphroditic
Yeah, they're
or
hermaphrodite, and they're also, which of these three terms on the left are they?
dioecious.
So if they have both reproductive organs at the same time, so every individual has both at the same time. Are all of the individuals, are there two different types, or are they all the same?
They're all the same.
Yeah, so what would that mean?
Oh, monoecious.
Yeah, perfect. Okay. Are you happy with this? Shall we move on?
Yeah.
Okay. So the last slide is oh no, I have the answer on the slide. What do I do? Okay, but this is just something that I added because he was mentioning it in the lecture, and I thought it was way too common knowledge to be like in a first year university lecture. Like you guys should have learned this a long time ago. So I just added it for that reason, but
Oh, yeah, I remember him talking about this.
yeah, I
Mm.
didn't even realize that. So these are all the possible types of contraceptive methods that Dr. Nelson listed, and I didn't even know that this was being used as like a contraceptive method. So this is sort of like the rhythm method means like when you track like the cycle of the female partner's ovulation,
Mm.
and like I was aware that people did that to maximize their chances of getting pregnant if they were trying to have a kid, but I didn't know that I hadn't considered that being used as
To minimize.
contraception. That seems so risky.
Yeah.
I don't know.
Oh my gosh.
Maybe I'm just uneducated. I don't know. But
I
yeah.
didn't know that either.
Yeah. Um,
What day would you like do this? Like,
Oh, okay. Yeah, I was just gonna say, okay, define like what would count as a prior method, but this is, you know this. I don't know. You know this. I'm not sure. I might post a message on Canvas. Here's what I'm gonna do. I'm gonna discuss with Tyson if it's reasonable to do like a final review session on Collaborate Ultra, or if maybe just give you guys slides or like what we want to do, because like I don't know how Tyson's been, because I've kind of already left like the ecology part to the last SL session.
Mhm.
I don't know if he's already covered that or not. Um, and if he hasn't, maybe he wants to do that on his Thursday session. So I'm gonna talk to Tyson and we'll figure something out, and then I'll let you guys know on a Canvas message. Um, I'm thinking like Wednesday, Thursday, Friday. It's gonna be like one of those days if I add an extra session, like if I move this session, sort of, so that I can have a final review session, or if I add an extra session to try to cover more detail on previous content. It'll be then. I'm not sure how realistic that is though.
Would it be like longer?
If I have a final review session, if we have a final review session, like if Tyson and I are doing that, that would definitely have to be longer than this, because we wouldn't be able to cover much otherwise.
Okay.
Um, yeah, I can't make any promises there though yet, so I'm not sure. If we don't have time for a final review session, I'm gonna put together some slides maybe and post them on Canvas so that you guys can practice with those, and I'll post the answers as well. Yeah, it'll probably be like some of the same questions from last time plus like extra questions for the new content. We'll see. Yeah, I just have like my lectures are gonna be like longer next week because all of my professors are doing like final sessions and stuff. So I don't know if it's realistic to try to schedule extra SL sessions. I don't know.
Yeah.
Tyson and try to figure that out. So last thing, what I like to do is like get feedback at the end. So like, what to do next time and then what went well. I usually do a drawing, but I'm just like, whilst you're writing, if you want to write anything.
um, the concepts, like an easy way to understand it.
What did you say?
Spelling this right.
So you can maybe write like, okay, thank you.
What did you say you were gonna cover next time?
So I'm gonna do the, like, I don't have time to do more detail on previous material. I need to cover the ecology stuff still. So if you've already watched those lectures and you know something in particular that you want to go over, you can add that.
Okay.
Thing in purple. You can't really see it in purple. I'm gonna add it in white.
I think that's it. Like, to really understand. I haven't really finished the lectures from ecology yet.
Yeah, that's okay. I mean, I'm gonna go over more than that, but yeah, we can
Okay.
take a look at that as well. Yeah. I'm sorry that you had to do this alone. I hope it was still helpful.
But that's fine.
Yeah, I'm kind of. We also had like a really low turnout last week, and I know like for some people, it's difference and stuff. But like, I'm sure some people were able to make it and just didn't, and I just like, it's just awkward for. I feel like bad that I feel like I'm interrogating like one person the entire time, and it's supposed to be like a group discussion. So I'm sorry about that. And
That's okay.
yeah, helpful.
Yeah, it was helpful. Thank you.
Okay, that's good. You guys, I'm gonna be posting this, so everyone who watches this. Why are you watching and you left your classmates alone to join the SL What else are you doing quarantined at home? Like, what else do you have going on? Like, hopefully we have more people next week. That's the last SL session. So, like,
What day is it? Sorry?
that's next week is the last SL session at some point. So join Tyson's SL session, join my SL session, because you only have to sit through one, and then we'll be happy. We'll be satisfied because we'll have had. Yeah. Okay. Yeah. Thank you.
Thank you.
Have a good weekend, and yeah, see you next week.
Thank you. Bye.
Bye.